Aberdeen, and there she is part of the TCAP program where she and her students are evaluating water use efficiency and nitrogen use efficiency in the National Small Grains Collection. Her presentation today will touch on many aspects of her work other than what I just mentioned. And with that, I'd like to welcome Dr. Chen and enjoy the presentation. Go ahead.
Okay. Thanks, Catherine, for the nice introduction. And one thing I wanted to correct, I'm an assistant professor, not an associate yet. Okay. So today I'm going to talk about the molecular breeding for biotic and abiotic resistance that we have been worked on in our breeding and genetics program. And wheat is an important crop in Idaho. It is the second largest cash crop in this state. The potato is the first largest cash crop. And wheat production in Idaho that ranks in the top 8 in the U.S. wheat. 50% of grain produced in Idaho State is for export. And 60% of the production in these states depends on irrigation. Based on the global climate change and water stress increased, so the water deficiency will threat Idaho high grain quality production. This is the Idaho map. In this map, the yellow area is the rain-fed area, and the green area is the irrigated area. This is the North Idaho, and this yellow area, the annual precipitation about 18 to 28 inches. And the yellow area in the south of states here, that is under irrigation. And the yellow area in the south, this is different from the yellow area in the north. And in this area, the rain-fed production depends on 10 to 18 inch of annual precipitation. University of Idaho has 2 breeding programs. One is on Moscow campus, and another is my breeding program in the south and southeast of Idaho. It's located here. And in the Moscow breeding program, the program focuses on soft-white winter 1 market class. And in my breeding program, we are working on several market classes. In the south area of the states, over 60% of Idaho wheat production from this region. And in this region, over 50% of production are for spring wheat. The major diseases and insects in these states, striped rust is the #1 important fungal disease. The dwarf bunt, crown rot. These 2 diseases are for high elevation and a long time snow-covered area. Fusium hydroblight is an emerging disease in southern Idaho, especially for irrigated spring wheat. Hessian fly is a disease usually, epidemic occurred in northern Idaho and Washington area. Abiotic stresses in these states include drought stress and drought and heat combined stresses. Outlines of my seminar today include 3 parts. The first part, I'm going to talk about QTL mapping of HTAP resistance to striped rust, green yield, Coleoptile and Root length, and the number of rows. Second part, I'm going to talk about marker-assisted pyramiding of resistance to Hessian-fly, physium head blight, and end-use quality. By the end, I'm going to share some preliminary results on association mapping of water and nitrogen use efficiency using national small grain collection materials that is supported by HTAP. The first part, QTL for HTAP resistance to stripe rust. Stripe rust is very important disease. Up to 80% of yield loss on highly susceptible cultivars. In 2003, the yield loss of 89 million bushels, and in 2005, 73 million bushels, and in 2010, 96 million bushels of wheat lost because of the stripe rust. There are two major types of the host resistance to stripe rust. The first type we call the all-stage, also called the seedling resistance. The second type we call the adult plant resistance, especially for high temperature adult plant resistance. HTAP resistance is an ideal trait for use of marker-assist selection, because this resistance type is risk non-specific and more durable. Compared to the seedling resistance, HTAP resistance is generally incomplete and controlled by many genes. Phenotyping of HTAP resistance is very expensive because the disease expressed at a late growth stage. It becomes effective when the daytime temperature is between 25 to 30 centigrade and nighttime above 10 centigrade. The mapping population used in this study consisted of 169 recombination inbred line derived from Rio Blanco by IDU 444. Rio Blanco is a hard white winter cultivar released by AgroPro. This cultivar is early maturity with RHTB1 dwarfing gene. IDU 444 is a released germplasm from my breeding program by previous breeder. This line has complex resistance to HTP, resistance to stripe rust, to dwarf bound, to snow mold. However, this line has okay end-use quality. And we used 732 markers, 96 SSR, 632 darts, and 4 STS markers, constructed whole genome-wide linkage maps using MapMaker for Macintosh. QTL analysis we used for computer program Mathemology QTX. Parents and recombination line were evaluated by infection type and disease severity at two growth stages, milk and soft dough, in two locations, MTV and Pullman at Washington in 2003 and 2004. The data was combined within homogeneous groups determined by Bartlett chi-square test. The linkage map we constructed, the average intensity is 66.7 centimorgan per marker. Like other published genetic maps, the D genome is less covered compared to A and B genome in this population. QTL we identified for disease severity a total of 4 QTL from 3 chromosome regions were identified that are associated with disease severity. The dataset showed here, the disease severity in milk stage and sulfur stage in the location Mount Vernon in both years are not significant. So we combined 4 data sets into one. It showed MTV all stages in both years. And another data set is from 2 growth stage collected from Pullman, Washington in 2004. So in this table, 4 QTL associated with the 2 data sets. You can see 2 QTL identified on 2B named as 2B1 and 2 significantly associated with data sets in MTV in both years and all stages. The LOD score is above 12, and the R-square that represents explained phenotype variation up to 31%. The QTL identified on 4A that is associated with datasets collected in both locations in both years that explained phenotype variation of 22%. The QTL on 3B associated with datasets collected in Pullman, Washington only in 2004. A total of 7 QTL were detected that associated with infection type. This slide showed 4 of them. This, again, the 2B1 and the 2B2, these 2 QTL associated with infection type data collected from MTV in both years. That explained up to 16% phenotype variation. The 4A QTL associated with infection type in both locations in both years. The 3B1 only associated with infection type in soft dose stage at a Pullman location. These 3 QTL also were detected associated infection type. The 1A associated only with the dataset from Pullman in 2003 explained 9% phenotype variation. The 4B associated with Mount in all stages in 2003 explained 7% phenotype variation. 5B associated with Mount in 2003 and 7%. This slide is a summary of the QTL for HTAP resistance. You can see from this graphic, the QTL on 2BS, 2B1, and 2B2 were significantly identified in all data sets of disease severity and infection type at MTV location that explained up to 31% phenotype variation. The 2 QTL were this study also confirmed the 2B1 QTL is located in the flanking region identified previously by other researchers in the resistant source, Cariga. The QTL on 2B2 identified in this study confirmed the previously published QTL in a similar flanking region in Luke and Springwheat, Louis. This tells us these 2 QTL are sound and probably can be used in the future marker-assisted selection. These slides showed the QTL identified on 4AL in this study confirmed the ones published in recent source Carrega. This, you can see the 4A was significantly associated with all dataset of disease severity and infection type in both locations. You can see these different colors and the notes here. And the QTL on 4A confirmed the ones in Carrega, and the markers linked to 2B showed in the previous slides, and these slides on 4A has been routinely used in my breeding program to genotype the parent lines we used in crossing.
Jianli, there's a few questions in the chat box if you would like to answer those now.
Okay, let me see. Okay, the question to Jamie Sherman, what type of QTL analysis we used, that is a composite mapping method. And the question to Sue is milk and soft dough stage, yes. And Catherine, the different growth stage, yes. What was the different growth stage again? The growth stage is in terms of like milk stage, that is after heading, the green start feeling, that is milk stage. Soft dough, that means the green feeling has already completed, and the green, you know, is kind of soft but not hard enough. And the disease data was collected by the collaborator, Dr. Jianli Chen, and they usually, because 2 locations, and they usually just read twice. And the first time they usually start to do milk stage, and the second is soft dough. And also because we are evaluating HTAP resistance, we have to wait in the late growth stage to let the gene to be fully expressed. Okay, and so these summary slides, and I have already talked in the previous 2 graphic. Then I'm go through to the next part of the QTL mapping for green yield in diverse environments. And this project is my student Jianli Zhang's dissertation project. I'm presenting some results from his study. We used the same mapping population between Rio Blanco and IDU 444. And this population, parents and recombinant inline line were evaluated in one of irrigated environment in 2006 in Aberdeen. And I put this in shorts, 06 AB. And the second environment we did in 2010 in 2 locations. One is in Aberdeen, one is in Blackfoot. The terminal drought stress defined here, that is water stress applied after heading dates. This population also were evaluated in 3 rain-fed locations, 1 year in 2007 in Rockland, 1 year in 2007 in Urban Valley, and 1 year in Rockland in Aberdeen, Rockland in 2011. So these rain-fed trials has the annual precipitation between 10 inch, 14 and 18 inch. And these slides showed you in Aberdeen, annual precipitation usually is below than 1 inch. That should be 1 inch. And the temperature from January to July is increased, and after July, the temperature decreased. And compared to the up graphic, you can see during the green filling stage, the terminal drought stress in South Idaho is very severe. If you don't have irrigation, and the treatment is like a rain-out shelter. And these slides showed you in 2007 in Aberdeen, we also evaluated partial of this mapping population under no water and with the drip irrigation. You can see this side, you can see some dehydrated plots showing up on this block, and these shows are all very green. And QTL, we identified from this population, these slides showed you 1B QTL showed consistently associated with yield in 4 of the 5 trials. There are 6 Aberdeen, there are 7 Urban Valley, there are 7 Rockland, there are 11 Rockland, and 2010 Blackfoot. You can see the 1B explained the phenotype variation up to 19%. And 3BS QTL is associated with the irrigated trial only. And 2B is associated with specific environments. And 3BL also related to specific environment. And 4BL related to the yield under terminal drought stress environment in both locations. And you can see here, this QTL, the tolerant allele from IDU 444, and these 3 QTL, the tolerant allele from Rio Blanco. And because the 1B and 3B are associated with irrigated trial, and we did test how the accumulated effect of these 2 QTL to the yields. From this slide, you can see the effect of 1BS and 3BS significantly greater than using 1BS only and 3BS only, and using 1 QTL of either of these 2 is significantly greater than don't use any QTL under irrigated production. And this slide showed you the accumulated effect of the 3 QTL under terminal drought stress. You can see from these slides accumulated effect of 1B, 3BL, and 4BL, and the accumulated effect between 1B and 4B is significantly greater than using single QTL and no QTL at all. And this 2 combination was not statistically significant different from above 2 and above 3, but is higher than the below 3 single QTL and non-QTL at all. To summary of mapping of yield QTL, and we concluded that chromosome 1B may contain a QTL associated with yield under different water environments. Second conclusion, result summary, we can see pyramiding of the 3 QTL on 1BS, 3BL, and 4BL significantly increased the yield under terminal droughts, while pyramiding of QTL 1B and 3B increased the yield under irrigated condition. The next, I'm going to talk about QTL mapping for coleoptile and root length. This is a study the visiting PhD student completed, and she graduated this spring. And this paper published in Crop Science. We used the same mapping population as previous 2 studies. This picture showed you IDU 444 has longer coleoptile, and this IDU 444 at both RHTB1 and D1 that contain the taller allele, and Rio Blanco has dwarfing allele at RHTB1 gene. And so we have a lot of study has been done previously for Coleoptile lens, but not many study has been done for root, especially combined Coleoptile with root in the same study. And from this study, we found IDU 444, the longest root of each seed is shorter than Rio Blanco, and the number of roots in each seedling is less than Rio Blanco. This my student developed the screening and phenotyping method. So she put the seeds on the germination paper and ramped up like this, put into the bucket. And each bucket we put same amount of water in the bucket. Then we put this bucket into the growth chamber. After 5 days, she take out the bucket and open up and read individual seeds and measure individual rods and the individual number of the rods. And the QTL she identified, and this slide tells you the common QTL on 4B. She identified that significantly associated with Coleoptile length and root numbers for both temperature. And the QTL also mapped in the flanking region of RHTB1 gene. And we mapped more QTLs for root numbers than the Coleoptile length and the root length. And you can see 4B QTL that can explain Coleoptile length up to 81% for the 67% of the phenotype variation. The heritability of Coleoptile length in this study is very high. The root length, the heritability is not as high as Coleoptile, but it's pretty good in in one of the temperature. And heritability for root numbers is pretty high. And because last slide showed the 4B flanking RTB1 gene, then we compared, how the effect of the RTB1 gene on the Coleoptile length and root length and number of roots. And using RTB1 gene to group the entire population into two groups, then we compared the two groups, get these slides. You can see from here, dwarfing allele, the lines with dwarfing allele under both temperature will significantly has shorter Coleoptile length compared to the lines with taller allele. However, the lines with dwarfing gene, they have longest, LR is longest root, and TRL is total of the root length. So you can see for these three traits, the lines with dwarfing allele has significantly longer root and more roots than the ones with the taller allele. This is a p-value, this is a difference. Then we also tested the effect of RHBB1 gene allele with the yield. You can see dwarfing allele, the lines with dwarfing allele has significantly higher yield under irrigation in 2011 under drip irrigation and under terminal drought environments in 2010 Aberdeen and 2010 Blackfoot. However, the effect of this gene is not significant on the 4 grain-fed trials in 2 different years. To summary of this study, we know, dolphin gene RHTB1 increase yield in hexaploid weights, but I didn't see the documentation showed on the genetic basis why the dolphin gene can increase yield. And this study kind of provided information. The dolphin gene increased the yield by increasing number of roots and root length. And from previous published research, we know dolphin gene reduce coleoptile length. That kind of the defect traits for planting dolphin lines under water-limited environments. However, in this study, we kind of provided the information, not necessarily dolphin lines cannot planted under water-limited environments if the roots characteristics is good enough that can balance the above ground with a short coleoptile length. And this increased number of roots and root length is the kind of new information derived from this study. The next, I'm going to show what we used for marker-assisted pyramiding. And this study, we used one of the known function QTL FHB1 located on wheat chromosome 3bs. We call the FHB1 gene. The best marker now published is UM10. The Hasenfly 25 gene that we introduced from one of wild species. The GWM610 tightly linked to this gene. And another known function marker we used that from George's program, that is the gene introduced from Agelopes monococca, the gene for high green protein tightly linked to YR36. And another QTL we identified not published, that from Rio Blanco by IDU 444 population, the QTL related to the baking volume located on 1A chromosome. The tightly linked marker is GWM33. And these parental lines we used in this pyramiding project. And you can see all of the 4 parents has the GLO-D1, that's 5 plus 10, high molecular weight glutini genes. The GLO-A3A is a low molecular weight glutini genes. The IDU 586 has, and Jefferson has. The other 2 doesn't have. So I'm going to show in the next few slides, through marker-assisted pyramiding, we obtained the progeny that combined these 4 target genes, and these 2 genes I'm not going to show here. So the objectives of this pyramiding study, we want to develop the hard white and hard red spring wheat with combined the 4 target gene. And also the second objective, we want to study the relationship between each and all of the introduced traits with yield. The third objective of this study, we want to develop breeding strategies using molecular markers. This is a pyramiding scheme we have applied. So we made the cross in 2009 and increased the seeds for F1 in greenhouse. And in these 3 crosses, and this cross we keep the 5F1, and this cross we keep the 10F1 and this we keep the 7 F1. For each F1, we planted 46 F2 seeds. The reason to do this is just for convenience for genotyping center using robots to do 384 PCR plates. So for each F1 plants, we collected 46 individual plants for DNA extraction. Then we send the plates to Washington Genotyping Center. So we grew 23 F1 plants from these 3 crops, and we got F2 individual heads. Then we planted in 2010 in 2010 in greenhouse individual plants, and 2011 we planted individual head rows in fields. And in 2012 we harvested head rows without any with the selection. In 2011 we have severe strep rust infection in the field, so we discarded some head rows. Like this cross, we kept 6, and this cross we selected 222, and this cross we selected 52 head rows, and 280 head rows selected from 2011 we planted in yield plots in 2012. Then we got this harvested. Then we will make selection to advance these lines into next spring to plant it in additional replicated trial. So from each generation in greenhouse, we collected individual F2s. We did marker selection and used the marker genotype and also field infection of strep rust and also SDS sedimentation quality data from lab. We made a selection and go advance the population into F4 in 2012. And this year, we basically evaluated for the phenotype in the field in terms of yield performance and adaptability. And we also send the same set of materials to Pullman, Washington, Dr. Jianli Chen's lab to do the field evaluation for HTAP resistance. And the partial of 280 lines, we send it to Jim Anderson in Minnesota for evaluation of FHB resistance, and the partial of 280 lines, we send it to Moscow for Hessian fly inside of the greenhouse screening. And we are currently doing the baking of this set of materials. And after this data obtained, we will plant around 180 lines in 2013. We will apply phenotype evaluation and marker selection and strep rust resistance and FHB and baking quality. And from this project, we learned, when marker assist selection should be used. Based on this, based on experience learned from this project, we would say from parental line to F2 using marker selection for target traits, and from F3 and F4 using phenotype selection for the other traits, and from F5 to F6 using both phenotype selection and marker assist selection. From this study, 45 out of 222 F4 we selected have 4 target genes, all QTL combined, and 30 of them showed good yield performance based on 2012's yield trials. Then the next study, I'm going to show some preliminary results for association mapping of yield and water and nitrogen use efficiency. This is my student Brian Bowman's dissertation study. The objective of our part of the project in the TCAP, and first we want to evaluate selected spring weight accessions for yield, water, and nitrogen use efficiency. Second objective, to develop high-throughput phenotyping models to predict yield, water and nitrogen use efficiency, use CSR indices in water and nitrogen-stressed environments. The third one is to identify novel SMP markers associated with yield, water and nitrogen use efficiency. The fourth objective is to identify novel accessions that can be used in variety development and improvement. This field looking the drip irrigation we have been using in 2011 and this year, and this early stage of wheat collection evaluation in last year, and this is the barley evaluation this year. This is the latest stage of wheat collection evaluation this year. And in these 5 years, our goal is going to evaluate 3,000 collection materials. 12 of them will be barley and 1,800 will be spring wheat. The last year spring wheat evaluation included 540 lines and 255 cultivars and breeding lines and 180 land races and 105 belong to unknown status. And these 540 accessions from 4 market classes. And last year, these 540 lines plus 60 checks, 600 total, evaluated under 3 environments. One environment with drip irrigation, normal nitrogen. One is drip irrigation, terminal drought and normal nitrogen, and terminal drought and low nitrogen. This year, we repeated 2 treatments because limited resource, and we repeated treatment 1 and treatment 2. And this is field nursery. The augmented field design we used, and last year we used 5 adapted checks identified from previous study, and as suggested by advisory board 2012 experiment, we used 3 adapted checks, same as last year, and we added 2 checks from collection materials. And these many CSR indices we evaluated, and these agronomic and physiological traits we evaluated. And the yellow highlighted traits that I'm going to talking about in the next few slides. And on this side of traits, I'm not going to talk today. And these slides use the 2012 data after we changed the CIGS grading number 4 to number 14. So the water index we used here is from grading number 14. The PRI is the radiation use efficiency. The RNDVI is the canopy photosynthetic area. And this index represent the chlorophyll A. The ONI indicated the nitrogen use efficiency. Okay, and someone asked a question. All of the CSR indices we used, we collected from JEDS. And these slides tell you 3 points. The first point, you can see NWI2 associated with yield only in the late growth stage. This is the beginning of grain filling. This the growth stage is heading days. So this is significantly associated with yield under in the late growth stage under irrigated treatment. And this is 18% was not significant. This is one point we can see from these slides. Second point, you can see from these slides, for these 4 indices you can look at together. The correlation value from both growth stage under irrigated environment is higher than the correlation value between these 4 indices with yield under terminal drought environments. This is the second points you can get from these. And the third points you can get from, you can look at the top 2 rows. And the indices, the correlation value of this index with yield collected from beginning rain filling stage is higher than the earlier stage. But this trend only shows under irrigation. But under terminal drought, you can see early stage R value a little bit higher than the late growth stage for these 3 indices. But these indices, the late stage a little bit higher than the early one, and this is higher than early stage. In barley, I don't have slides showed here. The barley is showed a little bit difference from wheat. And these are the 5 indices my students run stepwise multiple regression with yield. These are the 5 ones are important to develop the model to predict yield. So the model Brian developers based on the preliminary results. So using these 5 indices, we can predict about 50% of yield. And these 3 models we are going to develop and compare with the model of this one. And these 3, the difference between these 3 with the first one, we use the yield productivity change between treatment 1 with 2 and yield productivity change between treatment 1 with 3 and yield productivity change between treatment 2 and 3. We also will compare using the yield data that's calibrated based on the spatial variation, or we call the blow data or plot data. So we still have a long way to go to get a clear sense of how good these indices can predict yield. And this preliminary data that Brian obtained for association mapping, he found these many markers associated with traits of interest. This data he used from last year. One thing I want to point here of this one, he found 8 SMP markers that are significantly associated with yield productivity change and protein productivity change over treatment 1 and 2 and treatment 1 and 3. So these 8 SMP markers are more interesting, maybe related to water and nitrogen use efficiency. When we have more data, we will have clear results to present. And these slides I want to show, based on, this is from last year data analysis, and Brian haven't get combined of 2 years data yet. But from last year data, you can see the collection lines we named in the late maturity number 166. This one yield 145 bushels per acre under normal water and terminal drought environments. The yield is significantly higher than in treatment 1. And this line yield same very closely under treatment 3. So the yield higher from treatment 2 than treatment 1 indicated the high water use efficiency. The yield higher in treatment 3 than treatment 1 indicated combined water and nitrogen use efficiency. So this line we can see have both high water and high nitrogen use efficiency. And this line showed high yield on treatment 3, that is terminal drought and low nitrogen, than the terminal drought normal nitrogen. So we can see this line have high nitrogen use efficiency. And this line showed high yield in T3 than T2 and T1. So this one also we can see is probably have high water and high nitrogen use efficiency. So from previous slides, that's all about the molecular breeding that has been used and deployed in my breeding program. And in addition to this, and our breeding program has been trained for this is a college student from Idaho State University, this college student from Utah State University, this is a visiting PhD student. She did a Coleoptile and Root study. This is a visiting scholar. This is a visiting scholar. And this is a high school student from Aberdeen, Southern Idaho. And this is my student supported by National Need Fellowship. This is Jianli Chen, supported by University of Idaho Department Assistantship and Idaho Wheat Commission. This is Brian Bauman, supported by TCAP. This is Dr. Wang, supported by TCAP partially. And this is visiting scientist. And this is 3 students from Oregon State University, supported by National Need Fellowship. We provided training this summer. And this is the current team I have. But unfortunately, this senior technician passed away last week. And we will miss him. And this is the housing team this year. And these are the funding agency, Idaho Wheat Commission, previous CAP, Trixie CAP, National Lead Fellowship, BSF, Arcadia Bio Science, and Craft Nabisco. And these slides, I would like to acknowledge these many people, and maybe I missed someone. If you see someone missed, I apologize. And the first column are most for breeders in the U.S. The second column from USDA-ARS. And the third column are the extension people and professors. And the last column are the people from industry and growers collaboration. And with this end, I probably have 5 minutes to answer additional questions. Thank you very much.
Thanks, Jianli. And if anyone has any questions, go ahead and type those in now, and we'll answer as many as we can. In the meantime, while people are thinking of questions, I'd like to remind you that our next TCAP seminar series is not going to be until after Thanksgiving, so we have a few weeks of break, and our next presenter will be Dr. Pat Hayes, and his topic is Rapid Response in Breeding, the Challenges and Opportunities of a Shifting Landscape. So we will see all of you there. It looks like we might have a question coming up.
Okay. One question Sarah asked Did you measure CSR with TS or with different type? Well, I answered that question already.
Okay, great. Well, thank you, Jianli. We really appreciate you presenting your data, and have a great afternoon.
Thank you for everyone attending.